Hello everyone, welcome to tutorial 2. Seems like we have a good crowd. I'll give it another minute before we start. And so just to, yeah, you know what, I'll wait before we give the spiel. All right. So in this session, we will be looking at the EEG changes in health and depression. Sorry, one second. Okay, I'm actually going to pull Itai up for a bit of an introduction. Invite to screen.
Hey.
Hello.
Can you hear me?
Yep.
Okay. Your video minimized on purpose?
Yeah, I just shut my video off.
Okay, yeah. I just wanted to say before Frank takes over, hello everyone. Please feel free to stop us. This session is meant to be interactive. We have a certain program, but we're happy to answer any kind of question you have about the code, the way it's used. Frank made a very nice run through that helps people that have no clue about that kind of code. So it shouldn't be too daunting or confusing. But yeah, feel free. It's like a free form question as well, possible. And yeah, I think that's the thing I want to say. So have fun, and I'll let Frank take over.
Awesome. Thank you, Itai. Yeah, as Itai said, I sort of made this with the intent of, what I would have liked 5 years ago when I first started EEG analyses. So I definitely made that with, very beginner friendly. And yeah, please stop me. I'm going to change the structure of how I speak about things in this tutorial to allow you guys to have more time to ask questions and for more time to work on the depression task. So yeah, definitely I can see the chat. Actually, can you all give me a thumbs up so I know that I'm being heard loud and clear? You can see my screen, and we'll get started. Awesome, thanks, Taha. So first, let's just load in these packages here, and I'll give a quick. Awesome, thanks everyone, and I'll give a quick explanation. So NumPy, just using the arrays that we were talking about previously. SciPy is a really important toolbox that allows you, it comes with a lot of stats functions and signal processing functions. Matplotlib is just plotting things. And Foof is the tool that we'll be using to better characterize our EEG. I have I'll post the link here if you guys want to look at it later. But I'll be going to the website a little bit to show exactly what's happening. So these are just functions that we're going to be using. It looks a little daunting, but it's really just these two objects. It's a method to statistically summarize a single run and a group of EEG runs, or sorry, a group of runs, their EEGs. So a bit of background here, you will not be able to run this. It's just to show how we go from dipole to EEG. So from the, as Itai mentioned, our microcircuit simulations generate dipole data. What a dipole is it's a vector. And essentially, the vectors are summating, and you have this electric field, and that's what's being, you have the dipoles, which create an electric field, which is being measured by the EEG sensor. So I'm just going to show you quickly how we calculate the EEG. It's very simple. We use the four-sphere head model that Itai mentioned previously. So we have the four spheres here with different radii. We have different conductivity values for how the electrical, how the field travels through the media. We have the position of the dipole, so layer 2, 3, and we have the EEG sensor location. So all we do is we run through each simulation, and we add together the dipoles created by all of the populations. And then we simply use a function in LFPi to calculate the potential at a sensor that we put right on top of the head. And that's really what we're doing, what I'm doing in my research to create the EEG time series that we use for analyses. So if we scroll down, we're going to actually look at the dipoles and really appreciate how it's the pyramidal cells and it's their orientation that contribute to the EEG. So if we load this in, awesome. What we've loaded in is the pyramidal SST, PV, and VIP dipoles for one simulation, as well as the EEG for the healthy condition, 5 EEGs for the healthy condition. And well, I already plotted it, but essentially what we're seeing here is the Z component of the dipole, so the dipole that points up, all right? And you can think of the circuit as an X, Y, Z. So Z is pointing out of the skull, how your neurons would be oriented in vivo. And you can see the shape. So all of these are temporally aligned. You can see the pyramidal dipole moment, the SST, PV, and VIP. Notice the the y-axis here. Units are nanoamps per micrometer. What's it called? And the magnitude, of the pyramidal moment is about 10 times that of the other interneurons. This makes sense. The magnitude of the moment is created by the long apical dendrite that we see with pyramidal cells. Again, right? This, whereas all of the interneurons are quite circular, and you're not getting that massive current travel. So what we see is essentially the EEG measured at the scalp is a reflection of the pyramidal apical activity, right? Therefore, anything that affects this, the integration of these currents or the activity of pyramidal cells can affect the EEG. And what I mean by that is you see the SST and PV traces, for example, and it doesn't really look anything like the EEG. But what's important is that the SST interneurons are changing the activity of the pyramidal cells, which then is reflected in the EEG. So based on this, we can be motivated to look at the EEG as a marker for changes in cellular activity. And what's great about microcircuit modeling and realistic modeling in this case is that we're only changing one thing. We're changing SST inhibition. So it's we're more confident in saying, a specific change in the EEG between health and depression is a result of this one manipulation that we're doing to SST, as opposed to something in vivo where you have multiple different blockers acting on different channels or cellular knockout, which completely change the dynamics of a certain population. It's just a very precise way of asking a question. So we're going to be looking at how we actually characterize EEG. So if you look at EEG, you're going to see a series of waves, right? A series of waves. One wave with that looks like essentially a sine wave with a bunch of sine waves added to it, or a wave with a bunch of noise. And how we can characterize this wave is by collapsing the time domain and essentially saying or asking, what is this wave most like? What does this EEG wave correspond to the most? And how we do that is using a Welch's method for power spectral density. And I have a really good image for you guys here to show essentially what we're doing. So power spectral density plot, right, which is this, essentially you can think of it as how much of the EEG wave is similar to a pure sine wave at any point, or sorry, over time and on average. So for example, here we have the highest point as alpha, or 10 hertz, which means that this wave is very much like an alpha wave. In fact, if you were to create a bunch of sine waves and add them together with the relative proportions based on the power here, you'd actually be able to recreate the wave. So this is a great way to look at a wave on average and characterize it. It's a great way to characterize brain activity over time. So what a Welch function does is it essentially characterizes the EEG wave in little sections, as you can see here. So this EEG wave creates a PSD like this. This section creates a PSD like this. This section creates a PSD like this. And then you average these together and you get your final Welch function. And this example is good in another sense where you can see that there's a difference in the waveform itself here. So it doesn't look like there's any dominant frequency. Here we see a very clear oscillatory pattern at say alpha, 10 hertz. And you can see that's reflected in the EEG where there's a massive peak here. So from this EEG, from this Welch function or power spectral density plot, you can effectively capture this waveform just with this one graph. So I actually want to provide you guys with another link. That's really all we're going to get into with the math behind Welch's method and power spectral density generation. But I've just posted a YouTube link to a researcher his name is Mike X. Cohen. He does a lot of really great stuff on YouTube, and he'll be able to take this, take you much further into the math for EEG analyses or PSD generation. So is everyone following me so far? I hope that wasn't too mathy, but also gave you guys enough information to continue forward. So essentially, the PSD is just a characterization of the waveform. That's really it over time. Awesome. So for this, we're going to be using a Welch function, which is, and the Welch just means it's a method of averaging the PSDs created by it's just a method of generating a power spectral density plot. What we need for the function is the time series. So we're giving it this EEG. So this EEG object actually is a series of 5 EEG time series. I'll load that in, and okay, 5. So we have 5 time series here. So all we're doing here is we're saying the first EEG, look at the time series past 0.80,000. And why is 80,000 significant? Our sampling frequency is 0.025 milliseconds, which corresponds to 40,000 hertz. So what this means is just take the time series after 2 seconds. That's all. So we provide it with the time series of the data that we actually want to analyze. We have to provide the function with the sampling frequency. Then we define the window size. Here I've defined a window size of 3 seconds. There are different reasons why you'd want to have a larger or smaller window, but essentially the window size actually dictates how much frequency resolution you have for the PSD. So I have a printout here, and we see that with a window size of 3 seconds, we have a frequency resolution of 0.33 hertz. That means for every 0.33 hertz, we'll get a power. So we run that. Let's increase the frequency or the sample, the window size. Sorry, we'll have more data points. Therefore, we will have a smaller frequency resolution, and it's gone down to 0.2 hertz. If we put this to 1, we'll have less of a frequency resolution, and we're at 1 hertz. So it's a balance between your frequency resolution and your power resolution. I prefer 3 seconds for a window size. It just allows that 0.3 hertz is nice. And then the overlap is simply we're averaging multiple Psds, right, over the simulation. And this overlap just says, what's the step? This actually is a good example of an overlap of 50%. If the overlap was 0, this dotted line would be over here. And what the overlap does is it just accounts for the edge effects of using Welch's method. So everyone, you can just follow along, just run the cells, they should work. So all we're doing here is that we're just setting an end frequency for plotting. Essentially we're getting a PSD for I think 20,000. We'll probably get 20,000 frequencies, and it's just a large number to work with, and nobody really looks up to 20,000 hertz. So we're just setting an end bounds here just to make it a little bit easier to work with. And then we can plot. Okay, 1 sec, sorry. Where would title FS be? Oh, that's fun. Title FS is equal to 16. And FS is equal to 14. Should be in here somewhere. I just didn't run cell there. So here we have our PSD. As Itai mentioned, we didn't explicitly constrain the model to create this PSD, which reflects healthy human activity. Key characteristics are an alpha peak, i.e. the alpha power is high relative to all other frequencies. As well, the shape of the PSD plot follows a 1/f function, and what that means is, these two curves are the same data. It's just that the x and the y and x axes are log transformed. And if you were to plot a straight line here, as in 1/f, the PSD follows the same shape. The alpha peak and the PSD shape have been shown, since it's essentially a characterization of neural activity. It changes with consciousness, disease state. So there's a lot of rich information that you can gather from the PSD. But actually, is everyone okay with this explanation so far? Any questions, please don't hesitate to ask. Okay, seems like we're okay. So you're left with this PSD, and previously you would rely on expert characterization of the PSD. And what that means is somebody who's been looking at PSDs for a long time says, hey, this is an alpha peak. This is the 1/f slope. But it's very tough for a new researcher to say that they're an expert or have the the know-how and tools to actually characterize this PSD consistently and accurately. And that's what Wojtek, Brad Wojtek, I think his name is, created this toolbox called FOOF, which allows you to statistically summarize the PSD, and it will do that for as many PSDs as you give it, which allows you to just better compare different conditions. So for the healthy condition, we essentially give it the parameters of how big is how big do we allow a peak to be? How many peaks should we have in the power spectra? What's the peak threshold? And how are we modeling the aperiodic trend, right? So the aperiodic trend of the PSD is that 1/f function. So conceptually, you can think of the PSD as a combination of asynchronous 1/f activity and periodic activity that's resting on top of that. And that's what the that's what the FOOF model is explaining. So if you run that, we create the model, we set the frequencies that the model, the frequency bounds of the model. So we're only going to look at 1 to 30 hertz. And then this is the result. So we're looking in log-log space. So looking at this plot here. Essentially, what FOOF has created is, or what FOOF is describing the PSD is a straight line. Here you can see that it's using least errors estimation. Yeah, so we essentially have a straight line here, and then it models the peaks as Gaussians on top of that. And if we can see the Gaussians here as well, and the variables associated with the model. So with this model, you're able to characterize a PSD with the aperiodic components of the PSD as the offset and the exponents. So essentially the y intercept and the slope of the line in log-log space, and the peaks in the power spectra as their center frequency, their power, and their bandwidth. So essentially what that is you have a normal distribution, right, for the peaks. So you have the center frequency here, the band power, which is the height of the peak, and then the bandwidth, the essentially standard deviation of the peak. We also have some model parameters, the R-squared, and the fit error. And you can see that the model error, the R-squared is 0.93. That's a pretty good approximation of the raw PSD. So what we can do then is, from this PSD, we can take the aperiodic component of the PSD, and we can subtract that from the actual absolute power spectra that we have. And we can decompose the power spectra into its two components. Now, as opposed to assuming what is a periodic increase in activity versus an aperiodic increase, we actually explicitly separate those two to more specifically characterize them. So the aperiodic activity is this straight line here, and the periodic is here. Aperiodic activity, or the aperiodic of the power spectral density, is essentially a result of the power spectral density plot is, or the EEG is a result of these postsynaptic currents, right? If the cells are firing in an oscillatory manner, the currents will be oscillatory, and the PSD will follow a specific oscillation. But it's not very physiological, or healthy physiological conditions do not really exist where there's a entrainment of a specific oscillation at resting state. So the activity of the circuit will go from noisy, where the PSD isn't specifically oscillating at one frequency, to a little bit more organized. And that's what this periodic component of the PSD is. It's the periodic firing of the cells, or it's conceptualized as the periodic firing of the cells. And yeah, essentially, the periodic firing of the cells, which reflects the periodic component of the PSD, or the periodic processes which give rise to the power at specific frequencies. So we can do this again for the entire healthy condition. So we have 5 time series loaded. Same as before, we're just, for each time series in the healthy condition. We're going to calculate the PSD, and we're just going to trim it so that we're not dealing with these massive vectors. So we can see that we calculated the PSD here. We can check the shape. It's 5 by 300, which makes sense. 0.33 frequency resolution going to 100 hertz would give 300. We then calculate the mean and the standard deviation. And we can plot the resulting mean and standard deviation for the healthy condition here. So what we're doing here is, because I'll give a quick explanation because we'll be doing this later. We set the x value as frequency to the end frequency that we want. The y value of the plot is the power spectral mean for the healthy condition. The lower bounds of that shaded area represents the mean minus the standard deviation. So that's what we have here. And then the upper bounds is the mean plus the standard deviation. We then plot the mean, and we fill between the lower standard deviation bounds and the upper standard deviation bounds. We then create an inset plot with log-log axes, and we do the same. And that's the result here. So we can see that even over simulations, that wasn't something specific to or unique to that one simulation we saw previously. Most of the or all of the healthy simulations have an alpha peak. As well, all of the simulations have a 1/f type spectral decomposition. We can do the same for we can statistically characterize all of the PSDs in the healthy condition as well using FOOF, using the FOOF group function. So here, all it's doing is it's creating a model fit for each PSD that you give it, and it averages over those PSDs. It averages over the values of those PSDs, of those model fits, sorry, to give you group averaged fits. So we can run that, and here are the results of that. So we have 5 rows for 5 simulations, and you can see that for the first simulation, there's an offset of 15 and an exponent of 0.12, and you have the other ones here. And for example, the first simulation has a peak at 7 hertz, a peak at 10 hertz, and a peak at 14 hertz. And it does this for all simulations. So a very quick way to, again, consistently summarize your data, which is very important when looking at EEGs. We can see the group average here. So here's the first simulation that we saw previously, the fit, the aperiodic and periodic fit for simulation 2 and 3. And you can see the group averaged model fit and periodic component fit here as well. We can do the same thing for all simulations where we subtract the aperiodic. What is the standard deviation of the PSD plot for the non-group then? So this is the group, the healthy group power spectral density. This PSD is just one run, so there's no standard deviation here. There's no standard deviation here either. It's just the, what was the standard deviation? So this is just a way of conceptualizing what FOOF is doing. I didn't plot the standard deviation of the aperiodic fit and periodic fit here explicitly. We're actually gonna, I'm on my way to doing that. So we'll see that in a second. So to get the aperiodic and periodic standard deviation, we're going to actually recreate the aperiodic fit for each simulation. We're also going to recreate the periodic fit for each simulation. And then we'll calculate the mean periodic and mean aperiodic from the raw power spectra. So here we just, for example, use the gen aperiodic function, and we use the frequencies, the aperiodic parameters, and then the index of the simulation that we want. And we do that 5 times. Same thing for the periodic. We simply generate the absolute power spectra for each simulation, and then we subtract the aperiodic component from that absolute power spectra. We then average over all runs with standard deviation as well. And we just set up the plot as last time, where for each absolute, aperiodic, and periodic plot, we set the lower bounds and upper bounds for that standard deviation shading. And we plot it, and here we go. So this is the standard deviation that we were looking for previously. So we have the absolute power spectra here, the aperiodic component of the group average power spectra here, and the periodic component. So that's it for the explanation or the initial sort of showcase. We have task 1, which will be to compare healthy and depression PSDs. So the end result of this task will be to generate a plot like this. You'll have the healthy condition with the shaded area and the depression condition. So I've already, run everyone through the healthy condition. So for this task, you don't have to, reanalyze the healthy condition. As long as you've run through this notebook with me, you'll be able to do it. You'll have the variable saved. So the task will be to load in the healthy condition, the depression condition, calculate the power spectra, and average over the different runs. So we'll give 15 minutes to do this. And yeah, I will be here to answer any questions. Yeah, and I see a question. We have a question already. So I'll take up the answer at 3:30. And yeah, good luck, everyone. I will go on to answering the question now by Samar. So from PSD to waves, Ita, do you have anything to add there? Or?
Yeah, just a question. In this task that they're doing right now, is it only for the absolute PSD, or are they already breaking components? Just to clarify that aspect.
Sure, it's just the absolute PSD. Yeah, right, I can.
So,
yeah, it's just the absolute here.
Yeah, so good to clarify. All right.
Yeah. So if you all can reference that section of that section of the tutorial where I look at the average absolute PSDs for the healthy condition, and just apply that to the depression condition, and you'll be able to generate that plot. Yeah, that'll be it. So we'll take that up in 11 minutes. From Samar, from PSD to waves, is it possible to reproduce the EEG waves from the PSD? You lose track of the time. No. So this is very cool, Samar. Let me answer that. So this PSD, it's almost like you can think of it as like a recipe almost. So if you have you need to know how long the simulation is, but essentially what we're saying here is that the EEG that creates this wave, that creates this function, is 1 part 4 hertz to 2 parts 5 hertz to 1.2 parts 6 hertz. So it's really you can actually construct the original EEG by adding together a series of isolated sine waves. And this is actually people use this all the time to compress various forms of auditory data and things like that. A good example is in video or voice recording, right? say I'm looking at the audio recording for this example, or sorry, this tutorial, and there's a high-pitched ringing in the background. If I were to do a power spectral density plot on that audio file, you'd get all of the frequencies, and then you get this massive high hertz, say 20,000 hertz component that represents that high-pitched ringing. If I were to remove that component from the PSD, I'd be able to reconstruct my original audio without that component. So people go back and forth from the EEG to PSD all the time. So yeah, definitely you can do that.
That's an interesting question. I wonder though, isn't it more the spectrogram to EEG that will be able to reconstruct? Because you need to know when each frequency happened in a certain time, right? Otherwise, the PSD itself could have stemmed from a variety of, there can be the same combinations, but occurring in different times. Is that correct, Frank?
Yeah, it's a little bit more complicated, but it's you can definitely do it.
Yeah, I think the spectrogram, from the spectrogram, you can go to the EEG easily. Unless I'm mistaken, from the PSD, it's not necessarily a one-to-one, but you do have a lot of features that are captured there, right? Because the thing with the PSD, you abstract time out.
Yeah.
Right?
Yeah, I should have been a bit more explicit. So I can actually share this. So the spectrogram, as Itai said, is actually the way how you do it, where each of these, each time step is essentially a PSD, right? With the power represented by color. And from this, you can actually reconstruct the original signal. Yeah, you need to have, you are collapsing in the time domain, so you need to have a specific temporal bounds when you do it. But it's still definitely possible.
Yeah, and as Frank said, the spectrogram is exactly what people use in sounds. When they want to capture a chirp or a certain bell ringing, you can represent it entirely using a spectrogram. So it's pretty cool. All right, have fun doing the task.
So in the interest of time, I want to make sure that we have enough time for the other task. I will start taking up task 1. Here we go. So comparing healthy and depression Psds. So this essentially mirrored the healthy condition, where we're going to load in the depression data. So the depression EEG data was located in the depression folder labeled EEGD. So that's what I loaded it in as. And we're going to calculate the PSD for the depression condition. So I have the healthy condition here as well, just to show how it's mirrored and how you can grab that if you got stuck on loading the data in or anything like that. Essentially, what we're doing is for the depression condition, ensure that you're referencing the right array, right? So we have the healthy time series here, and we have the depression time series here. And we're going to create a PSD using the same parameters. So 40,000 sampling frequency, window size of 3 seconds, and an overlap of 50%. You can do that for both. There we go. Here's just a quick check to make sure that everything's loaded in properly and you didn't change the variables at all between conditions. Sorry. So 5 runs with 300 data points for the healthy, 5 runs and 300 data points for the depression condition. Now, all we're going to do here is calculate the mean and standard deviation for each condition. So I have a bit of a typo in my final copy, but that's fine. What we're doing is we have this array, right, of 5 by 300. And we're going to have with each column representing a frequency, right? So to get the mean PSD, we're going to average over each column, i.e. average over each frequency, and we'll get the mean frequency power at the mean frequency power and the standard deviation. We're just going to use NumPy here. So the depression power spectral mean is equal to NumPy.mean of the power spectra over all runs, and we're going to average over the first axis. So can everyone give me a thumbs up for this? Has everyone gotten to this point? Are they stuck on the actual calculation of the PSDs? Because this is with the other section sort of depends on this being loaded in properly. So I just want to make sure everyone has a chance to continue with the rest of the tutorial. Awesome. Oh, sorry about that. I don't know. Maybe my internet connection dropped there for a second. Can everyone still hear me?
Yes,
Awesome.
You dropped for a sec, but you're back on.
Great. Okay. So I see 1 thumbs up. Is everyone else doing all right? Or I can go over loading in and all of that stuff so that everyone can follow along. Okay, here we go. Yeah, so run the whole notebook. So essentially from, well, you know what, actually. Let's see. Martin, good. Sure. Okay, so Samar, what I want you to do is, you can run the whole notebook, but an easier solution would be to just copy this end freak line and put it after in here, right? So we moved the line from here into here. And do that for the, the depression condition. It probably ran for everyone else because, yeah, you can run the whole notebook and it will save that frequency variable from previously. But if you're running it in isolation, yeah, do what I just did. So this end freak line will be up here. Just put it in here. So we're going to run that.
Frank, I'm not sure if it's just me, but I see half of the screen your video and half of the screen the code. So
okay.
Yeah.
Here we go. This should.
Yeah.
Awesome.
That's better.
Okay. So I'll just reiterate what I said so that everyone's on the same page. So what I assume everyone's doing. Yeah, this should work. So you're going to copy this. And ensure that the end freak is inside this for loop, specifically after you create the frequency variable, and it will run. All that's happening there is that you're referencing the variable before you create it. So check the shape, and then you're going to calculate the mean and standard deviation over the first axes. Okay, so it seems like everyone's okay. And for the plotting, what we're doing is, so you guys should have seen essentially this, or perhaps this, sorry. Yeah, this, and this would be empty. So we're setting depression X the same as healthy X, which is the frequency to the end frequency index. We're setting Y to the mean, whatever variable you've chosen, every variable name you've chosen, as long as it's the mean, which you've created up here. We're setting the lower bounds for the shaded portion as the mean minus the standard deviation, and the upper bounds as the mean plus the standard deviation. And that should be it. You shouldn't have touched anything over here. I set it up so that you just need to provide the proper values, and it should run. So after running that, you should get a plot that is identical to this. So can everyone give me a thumbs up if they're there? Maybe there's a lag on like the chat or something like that. Because usually I'll ask for a thumbs up and then I get it maybe 2 minutes after. So maybe Crowdcast is doing something, but if you followed the instructions, you should be here. So let's characterize this PSD. We can see that in the healthy, which is in black, versus the depression condition, which is in magenta, with standard deviation indicated by these shaded areas. You see an increase in theta and alpha activity, as well as beta, and then a small broadband increase. And you can see this as well in the log plot. So many clinicians will use this as a biomarker for the condition. So say depression in this case, you can take the difference in power and say that a biomarker for depression, or SST-mediated depression in this case, is an increase in theta, alpha, and beta power. But what we're going to do is be a little bit more specific with our approach And we're now going to decompose this absolute power spectra into its aperiodic and periodic components. And I might have forgot to mention this before, but this is a very powerful tool because essentially what it allows a clinician to do, or what it allows you to do, we have the luxury of testing our periodic measures against the actual network itself. But by decomposing the aperiodic and periodic components, you're able to gain insight on specific functions. So for example, I mentioned before how the periodic reflects the oscillatory activity of the network. The aperiodic activity can give you insight on things such as firing rate or overall network activity and things like that. So we'll get into that a little bit later after you've decomposed this. But this is actually a very it's fairly novel and fairly powerful as well. So the second task, therefore, will be to decompose this spectra into its periodic and aperiodic components. So essentially what we'll be doing is part 4 up here, but applied to the depression condition. So you should get a plot here, which compares the healthy and depression, and all the way down here, where you'll have the healthy and depression absolutely, and then their aperiodic and periodic components. So I'll give you guys another 10 minutes to do that, and we will check in. We will check in at 3:50. And is there any are there any questions so far? Any if anyone's interested in anything a little bit more in depth, I can talk about that now. If they're still struggling with maybe something I said before that I glanced over that you were hoping that I explained a little bit more, we could talk. Really anything. I'm hoping for some discussion, some interaction with you guys. So. I guess Itai, if you have anything else that you think that I've missed or hoping for me to explain a little bit more, I can do that in the meantime while people are thinking of questions or.
Yeah, I think you really covered things excellently. I don't remember if you said it specifically, but most of this analysis is fairly simple when you think about it. The simulations are heavy, right? You have all those models. It took ages to do that. But then that you're ready to simulate the circuit and get EEG. And obviously there are many things to learn about how to do it right. But as I hope you see in the code, it's not a long code. It's also not so complicated to parcel out. So yeah, I mean, I think a researcher has always the option of taking existing models, using those tools that can simulate them, get EEG and ask questions about it, and then relate it to experiments. So that's something to keep in mind. Whatever your background is and however deep you want to delve into the biophysics, yeah, I mean, simulating EEG from detailed models is possible. And those tools actually took a lot of effort by those groups to write, but they're available now. So essentially, they're ready and open for the public to use.
Mm-hmm. And just in case people missed it before, I'll link right now the LFPI toolbox, the Mike Cohen, or the EEG PSD walkthrough videos that I referenced before, as well as the FOOF toolbox. They offer a lot of really great tutorials that go into much more depth than we are here. We're sort of providing a more holistic view from start to end. Yeah, hopefully, definitely there's a lot of really interesting stuff in these links. So feel free to utilize it.
And in general, the advantage of computer simulations, and people have done it when they simulate the plane before they build it, is that they enable you to test things before you sacrifice either animals or resources. And that's exactly what you would do here. You can ask questions and test them. And the analysis here is really the same method that you would use in the real brain data. So, yeah, I mean, it's interesting, I think, and we see it as well that Frank in a subsequent project just is analyzing actually real data in a very interesting EEG experiments using the same methods. So this is really, in a nutshell, it's like a virtual experiment here, and that's a pretty cool thing.
Definitely, it allows you to. It's very hard to test certain things in vivo, right? Specifically with EEG, people are trying to relate it to, the microscopic changes in specific channels and things like that. And by using these virtual experiments, as Itai mentioned, with the actual human EEG that I'm looking at now, you can take those insights and apply it to new things, right? So you can check, changes in, synapse parameters, perhaps, and look at the change in the PSD and be a little bit more confident in interpreting the PSD of a, real or awake living person. So there's a lot of utility in this. It allows you to really parameterize the functional components of the PSD and things like that and apply it to novel situations. So it's a great learning tool to see how different things affect the electrical activity of the brain and stuff like that. And I didn't mention before, but we're simulating a network of 1,000 neurons, but it's not too far. It's not very tough to simulate a network of even 100 neurons on your laptop, specifically like with the detail that we do. So don't think that you're losing out if you don't have access to like a supercomputer or things like that. Yeah, about 100 neurons I've done on my laptop previously. So if you reference back to the circuit code that I showed before in session 0 or session 1, sorry, you can look at the EEG of hundreds of neurons and gain some insight there as well. All right. So this task, can sort of be broken up into 2 subtasks, essentially, with the second being a little bit more difficult than the first. So I'm actually going to start now. I'll go into, the first half of this task, and hopefully, if anyone's struggling, they can use this example and keep on working into the second. And then I'll give a little bit more time for the second as well. So here we're going to be comparing the healthy and depression Fuchs models, which is essentially just this statistical summarization, the statistical summary of the PSDs in the healthy and depression conditions. So a little bit redundant, it is redundant, but in case anyone didn't get a chance to finish the past task or the previous task, we're going to start by loading in the depression data, and we're going to again calculate the PSD for each condition, or sorry, for each run in the depression condition. And then we're going to set the start frequency and end frequency that we're going to model the, let me see if I can zoom this in a little bit. There we go. We're going to set the start frequency index and the end frequency index for the model. And we're going to set up the other parameters for the model, such as the number of widths, the peak width limits, and the peak threshold. So these variables essentially allow you to tune the model to make sure that you're not overcounting specific peaks in the power spectra or ignoring other ones. So it doesn't interpret the PSDs for you. You need to have a hand in that as well. So yeah, you need to make sure that you're interacting with the model. But I've looked at this data before, and 3 to 30 is fine with these parameters. So we're going to fit the model with frequencies from the start index to the end frequency index. And we are going to look at the power spectra. And this section might have given people some trouble, but here we've created an array of the power spectra, the depression power spectra, right? So if we do the EPS shape, it's five columns by five rows by 300 columns essentially, which is with each column being a frequency. And what we're going to do is take, this is just an indexing method where you take all of the rows and the columns from the start freq index to the end frequency index. So there's a question here. Yep, so setting a peak, the question is, sorry, let me start answering actually, there we go. So the question is, wouldn't setting a peak width affect the bandwidth estimation? And yes, that's true. But physiologically, if you're thinking of the bandwidth for a specific functional oscillation, it's very rare that an oscillation functionally is working over 12 hertz, right? That's if you're going from 10 to 20, that's almost doubling. That is doubling the activity of the oscillatory activity of the neurons. So yes, you are limiting the bandwidth, but functionally, the bandwidth of a specific oscillatory process, I've never seen an example where it's gone over 12 hertz. Mostly this is used for if you have a broadband peak, you're not setting that broadband peak to like 30 or 40 hertz. It's a constraint on the model to better separate the periodic and periodic. Yeah, it's a priori. Yeah, it's an a priori parameter, but you can play around with it and see if that affects it. It's specifically it's important when you're looking at, or this is a good example actually, when you're looking at the power spectra of like a fairly high frequency resolution. So for example, if you look at the depression condition here, assuming that pretend that this is 1 PSD, if I set the bandwidth to 1 hertz or something like that, the minimum bandwidth to 1 hertz, the model may model this frequency as 1 peak, this is 1 peak, this is 1 peak, and this is 1 peak. When, more often than not, people would say, no, you have 1, theta peak, 1 alpha peak. So you don't want the model, it's essentially to prevent your model from overfitting certain peaks. That's where it's mostly used. So I hope that was a good answer for you. I'll provide my email in the chat later. Awesome. Okay. If anyone has any further questions or questions about the code, just email me and I'll be happy to answer anything. So for the depression condition, we print out these parameters and the aperiodic and periodic parameters. And we simply are going to plot the simulation fits as well as the group fits. So first row is healthy, second row is depression. And you can see, just comparing these two, comparing the first row versus second, that it seems like the depression have an increase in theta. Okay, but and when you average over the group, you can see that the group modeled theta is much higher in the in depression compared to healthy, as well as an increase in low beta. Right, and to actually look at that with respect to the raw PSD, we'll do the same thing that I did before, where you generate the aperiodic component of each run's PSD, and you subtract that from the absolute component to separate the periodic, aperiodic, and absolute. So we do that here. Again, this just models what I was doing previously in the healthy condition. We set the plotting parameters for the shading for each absolute periodic and aperiodic and periodic plot. And this should all fill in. This should be all filled in for everyone. And this is the resulting plot. So we're going to spend a little bit of time on this, but we can see that if you're just stopping at the absolute power spectral characterization between conditions, you'd say that there's an increase in theta and alpha in depression, as well as a small broadband increase or something. But by separating these components, you can now say you can characterize components of the PSD or sections of the PSD with much higher accuracy and precision. Just an overall, I'd say, more sophisticated analysis of the PSD. So here we see that as opposed to a small broadband increase, you have a fairly substantial broadband increase, specifically the aperiodic component of the PSD, which increases with frequency. So that's observation 1. Observation 2 is you have a periodic increase in theta and low beta, no periodic increase for the broadband component, and no periodic increase in alpha. Let's see, make sure there's no questions. Perfect. What's interesting here is that the aperiodic component of the PSD reflects, as I said before, non-oscillatory components of cellular activity. And what we saw previously with the raster analysis was that depression, the depression condition that we modeled using a decrease in SST-mediated inhibition, increased the baseline firing, increased the noise of those microcircuits. And it's very clearly showcased here in the aperiodic component. What's cool there is that we've connected that, but now other clinicians and you all, if you want to take this and apply it to your PSD analysis, by doing this, we can say that the aperiodic component of the PSD reflects this aspect of circuit activity fairly well. So by decomposing the absolute PSD into its aperiodic and periodic components, can gain further insight into the different processes of the network you're analyzing. As well, you can see that as opposed to a large increase in theta alpha. It's a small increase in theta and a small increase in low beta. And we haven't shown this in the tutorial, but if you look at actually the rhythmicity of the populations with respect to their tuning to firing at a specific frequency, you'll actually see that in the depression condition, depression circuits oscillate a little bit. Healthy condition is oscillating fairly consistently at alpha, and the depression condition decreases its selectivity essentially. And this is mirrored, very well in the periodic PSD. So again, this better allows you to make connections between macroscopic phenomena that you can measure in a clinic after a 30-minute EEG session, and these microcircuit signals that you would realistically, it would be very difficult to access in vivo. So I hope everyone has been able to. I hope everyone they're able to generate these plots. We have a couple minutes left, so I'm just going to go through just like a quick section that we had in case there was any time left. But we can also use these parameters that we generated here and model these parameters specifically. So here we can separate peaks in the theta, alpha, and beta, and this can just, it's a little bit easier to see the difference in depression versus healthy, how it's present for theta. There's no difference in the two groups for alpha, and you can see an increase in beta power in depression versus healthy, a little bit easier there. We can also do the same for the aperiodic measurements, and for both of these, there's a significant difference between offset and as well as exponent. Exponent, again, is just the flatness of that line, which is also related to different synaptic properties like excitation and inhibition balance. So I hope from this tutorial and the last tutorial, everyone's gained some insight on spike analysis and EEG analysis, and how by using microcircuits and by using this methodology, you can gain insight on multi-scale mechanisms of, here we use depression, Itai mentioned schizophrenia markers, as well as you can apply this to any sort of disorder, neurological disorder, and really parse out specific relationships and biomarkers that are very difficult to study in vivo. So again, we're out of time, but if anyone has any further questions, I'll type my name out in the chat or my email out. But
just to clarify, we technically have 15 minutes.
Oh, really? Oh.
Yeah.
Oh, okay. Awesome. I was freaking out a little bit there, trying to finish up quickly. Oh, sweet. So we can go through it a little bit more in depth then, if anyone has any further questions, or we can go through the bonus extra section. What do you think, Itai?
I think we can give them a few minutes to decide and come up with questions. That's definitely a good option of how to use the time. And if there aren't any questions in a few minutes, you can go through whatever you want. Yeah, because I'm pretty sure, right, it was 1 to 2:30 and then 2:45 to 4:15, so you're good. And you have a few questions, so yeah.
Awesome. I don't know, I must have, I saw 4 o'clock somewhere.
Yeah, originally that was the plan, but we've extended it to be like the other tutorials.
Oh, I see. So yeah, if anyone has any questions, you can ask it in the ask a question section or in the chat. I can provide my email now. People are writing questions.
We're happy to hear also maybe what you were hoping to get or to implement in the future from this. So if you want to even just as a sentence share, if you feel like you might be using, what do you do with EEG maybe or anything, or then feel free to post that. In an in-person meeting, obviously, we would be seeing each other and, you can raise a hand and talk. Technically, we can always call you to, chat on stage. So that's also a possibility if you feel like. So yeah, let us know.
So Samara, I'll start answering. Is it possible statistically to use the same method to compare Psds for the same individuals or group, for example, Alzheimer's disease, before and after a specific intervention? Yeah, definitely. You just have pre and post, essentially. And indeed, like if, for example, we were to model some sort of treatment here, we would take the depression microcircuits, change some sort of variable, and compare the PSDs before and after. So it's not specific to two different conditions. It's just an arbitrary grouping of your data. You can also use this, I've shown it with respect to resting state activity, but you can also use this for event-related potential, ERP analysis. Yeah, really trying to think of an EEG measure that wouldn't do well with this, but it's essentially an added tool that you can use whenever you're doing a PSD or PSD analysis. So yeah, definitely. And if you need any further guidance on that, you can always email me and we can get into it a little bit further. Another great question. Can we see this research heading towards a cortical cortical micro mini colon modeling? Yes, definitely. I'm really interested in that, in doing that in the future. So yeah, yes, we can foresee that. I don't know, Itai, if you have anything else to add to that, but yeah.
Yeah, I just want to make sure I got the full question correct. Yeah, definitely. I mean, I think one of the things you can ask at the level of a single cortical microcircuit can involve those loops. People like John Griffiths look into that actually, and all in a little simplified representation, and usually more nodes. But you could also look at it in terms of a single circuit, and we would be really interested actually in getting some insights from the detailed simulations of the the thalamic cortical thalamic loops, right? And then maybe having a better idea of how the simplified models, what kind of properties they should capture, right? So yeah, totally. It's there is actually a lot that is known about it, particular layers that connect back to the particular regions of the thalamus, and yeah. I believe, if I'm not mistaken, that Dr. Sean Hill has done a bit of work about some of it, and the cortical, like the thalamic cortical interactions with detailed models. But yeah, hope I'm not misremembering, but I'm pretty sure that he's done that. So yeah, it should be very interesting. It would be worth commenting also, as you see, that a lot of the work can be done on Python these days. That wasn't the case back when I was doing my PhD. It was just Neuron, really, but they made a good move to really put a lot of the things to interface with Python. And yeah, I mean, that makes it much more accessible to other researchers, right? So yeah, you can see it even in the tutorials that you don't require so much of a dedicated software to run those things.
There's also the visual aspect of Jupyter notebooks as well that we really appreciate. You know, it's good to be able to, you know, have scripts and pipelines that pull, you know, from time series to outputting a PSD. But the process of actually getting there, Jupyter notebooks are very useful in that sense. You know,
Yes.
yeah.
And Neuron was never a visualizing software, really. That was never its strong suit. But yeah, now you can really plot and take advantage of all those functions to compute PSDs. You do it all in one language, essentially. And an interesting comment also, maybe to add to that question about conditions pre and post, you can totally do it, and even a lot of the depression data is like that, right? You have a treatment, you have a group before the treatment, during and after, and you have then subgroups about the ones that responded well, the ones that didn't, right? So the temporal aspect of, you know, and the ability to calculate PSDs in each stage is super relevant, and there is actually data that you can constrain the models with. So that's a very nice question.
Actually, Samar, if you look at, I can send the paper in the chat, but the group, the Wojtek's group, which created FOOF, actually, I think, tested it on an Alzheimer's database. So you have a great example there. John comments that Neuron's GUI looks like it was made in the Stone Age in the 90s. Thanks, John. I guess I can ask again the chat a question. You know, last year we looked at actually simulating this code on your laptop or on SciNet. This year we focused a little bit more on the analysis and pulling information from those simulations. I see some familiar faces from last year, and you know, we're planning on doing this again into the future. What were people hoping for, and what are they looking to see? Are they interested in seeing in the future? That would be really helpful, so that we could, tailor the experience to what you guys find interesting. I've also posted that that FOOF paper I mentioned before in the chat, if anyone wants to take a look.
Yeah, and the feedback that Frank was asking, feel free to provide in the form after. There are the surveys. We're happy to hear any recommendations. The virtual interface doesn't always allow the presenter to get the vibe at any point of time. But yeah, it was nice that quite a few of you followed and asked questions and tried and succeeded for the most part. So that's very nice. And yeah, feel free to provide feedback. I think we are drawing to a close. Frank, you did a marvelous job. Thank you. Last year we were sort of together on that, but like there is really no need for a second person here. So that was lovely. Any last words? I'll leave for you.
No, nothing really from me. Yeah, I just wanted to thank everyone for attending. It's the summer school is it's a behemoth of a lesson plan, but it's very valuable. I don't know of many other places that take you from genes all the way up to modeling whole person interaction. So I hope the microcircuit level section was handled well. You enjoyed it, and I hope we provided you with insight that you can use tomorrow in the whole brain modeling section and thinking about these micro scale interactions and how you can generalize that to whole networks and interactions between regions. Yeah, thank you all. Yeah, and we'll see you tomorrow then. Take care, everyone.